T cells, and basically with all the other types of T cells, whenever I map these out, I map them out based off of the things that affect their differentiation and then things that contribute to their effector function. So for the differentiation, obviously, like I had mentioned before, this is totally reversible, and it's dependent upon the tissue where the dendritic cell came from, the innate immunity, and then also just kind of the nature of the pathogen itself. But I'm just going to go ahead and just branch this off based off of the cytokines that are involved in the differentiation, and then the master regulator transcription factor. So for the two cytokines that are involved in the differentiation process, the ones that I'm going to list are just interleukin 12 and then interferon gamma. And these two guys come together to result in the production of the transcription factor known as Tbet. And as far as where these two cytokines come from in terms of the innate immune system, for interleukin 12 we get most of that through dendritic cells and then macrophages, and then interferon gamma is something that we get from natural killer cells. Which, when you think about the effector function of Th1 CD4 cells, that makes sense, right? So once we start to produce Tbet, issue, or it's not really an issue, but a phenomenon of polarization can happen with Th1 CD4 T cells, because the cytokines that also induce their own differentiation also serve as effector functions. It results in the production of cytokines that play a role in promoting their effector functions that also induce differentiation, that cause a role in effector function, and so on and so forth. So this quickly can get out of hand by the means of a positive feedback loop. This results in the very rapid expansion of Th1 helper T cells. Alright, so let's talk about their effector function. Before I do that, mentioning the fact that their differentiation is completely reversible. So for the effector functions, we're really dealing with intracellular pathogens, and the unique thing about all helper T cells is that, and cytotoxic T cells for that matter, is the fact that the cytokines are made after contact. It's the fact that they do this that really kind of is their weak spot, I think, in terms of how long it can things that can fail whenever they try to attack a pathogen. So there's many things that they can do to fight intracellular pathogens, and the best understood mechanism that they have is macrophage activation. And then the two cytokines that play a role in macrophage activations are the CD40 ligand, so remember that one is membrane bound, and then interferon gamma, which is soluble. And as you can imagine, this causes an increase in the antimicrobial activity, including but not limited to increased production of intracellular proteases, increased production of oxygen radicals, and increased production of, or just increased, production of the generalized mechanisms that you know of that help macrophages fight intracellular pathogens. Right, so that's it for macrophage activation. Next thing that I wanted to mention would be the fact that it's able to kill the chronically infected macrophages, so that they will release the intracellular pathogens that are hiding inside of them to be destroyed by the immune system. And the two cytokines that are involved in doing this would be the Fas ligand and then tumor necrosis factor beta. And in case you don't remember that from when we talked about the cytotoxic T cells, the Fas ligand is something that's membrane bound, and then the tumor necrosis factor beta is the soluble secreted cytokine. And the reason we do this is because we want to expose those pathogens that are hiding inside the macrophages to the other healthy immune cells in the surrounding areas. Next thing, you should be pretty familiarized with this, but they're helper T cells, so they're going to play a role in T cell proliferation. And this is accomplished by the action of interleukin 2, which is otherwise known as the T cell growth factor. Next thing is they also play a role in macrophage differentiation, and we didn't go in much detail with the mechanisms regarding this. I'm just going to list interleukin 3 because I think that's the only thing that we've talked about as far as that goes. We didn't really talk about so much the other interactions that are involved in that. The next thing is really just inflammation, and then macrophage homing or leukocyte homing, if you want to think about it. And this is accomplished with CXCL2, which is the common chemokine for macrophages.